Hello everyone and welcome to this lecture on diffusion, osmosis, tonicity, and osmolarity. So the topics of review is kind of the title of this lecture. So we will discuss diffusion, osmosis, tonicity, and osmolarity. And I'm sure a lot of you have already covered these topics in your previous classes, but I wanted to make sure that we were all on an even playing field. So let's get started with diffusion first. This is when we have a net movement of a substance from an area of a greater concentration to an area of lesser concentration. So we could see this in the example down over here as this person is putting in some type of dye into a solution of water. We're starting to see that those molecules and ions are constantly moving due to their kinetic energy, and this area where we first put that dye into is the high concentration area. So from there, we're going to move into the areas of lower concentration, and as long as it's unopposed, we will see that diffusion is continuing until the substance reaches equilibrium. And all of these molecules, as you can see in here, are evenly distributed. So let's continue to talk about the rate of diffusion. And so we have these two categories, the steepness of the concentration gradient and the temperature. So when we're talking about our steepness, we're saying that when we have a larger concentration in one side versus the other, which is low concentration here, we see that there's a faster rate of diffusion. Whereas if they are closer to equilibrium, then we have a slower rate of diffusion. So you can see it's less steep here using our example of the slide. Then when it comes to our temperature, if we have higher temperatures, then that means that we have more kinetic energy or more random movement. And so when we, for instance, put a tea bag in a hot glass of water, we are going to see that the rate of diffusion is going to be faster versus if we put a tea bag into a cold cup of water. All right. So we have different types of diffusion. We are just going to talk about simple diffusion here in regards to a cell. So here we are seeing our plasma membrane, our border. We can see that phospholipid bilayer, and this is representing the outside of our cell, and this is the inside of our cell where our cytosol can be found. And here we see our molecules are passing between these phospholipid molecules, and our smaller nonpolar solutes are going to be easily moved through diffusion down the concentration gradient. So again, going from a high concentration area to a lower concentration area. So areas in our body where we see this happening is our respiratory gases. So when we have that capillary bringing in that fresh oxygenated blood, it will then move the oxygen into the tissues and pick up the carbon dioxide. And also we see this with some fatty acids, ethanol, and urea. This is not really regulated by the plasma membrane. Again, we're focusing on the concentration gradient here. Now let's talk about facilitated diffusion. We have two different types. So what we saw previously was simple diffusion, where that molecule is just simply moving through the plasma membrane. And then we have our two types of facilitated diffusion, in which we are going to use some proteins pictured here to assist us in doing that. So let's start off with our channel-mediated diffusion, our first type of facilitated diffusion. This is when we move small ions through a water-filled protein. So our channels here have to be specific for the ion type. We do have two different types of these channels. We have leakage channels, which you could see here just simply are going to move through. These again are going to use our concentration gradient. So we see that we have a high concentration down here, so we move them out of the cell. They will always be open and again use this gradient for kind of their type of regulation. And then we have our gated channels. So the one I have pictured here is a voltage gated channels. You might remember discussing this in the nervous system or the muscular system. These will typically be closed and will change depending on the voltage of the cell. So let's say we have the inside of the cell, usually our resting membrane potential is going to be negative. So if it starts to become more positive, it reaches a threshold, and then it is going to open up and allow the ions to move through. We can say these are like our sodium ions moving into the cell, making it even more positive for like an action potential to take place, for example. Let's continue to our second type of facilitated diffusion. This would be a carrier-mediated diffusion, where we have smaller polar molecules. They need a carrier protein to move them through. So here we could see the example of glucose, and it is going to go to its specific carrier-mediated protein, and we are going to attach, and then we get a conformation change in the protein, and that allows it to move the substance, in this case glucose, through down its concentration gradient. Again, higher concentration outside, so we are moving it inside where we have lower concentration. We also call these uniport or uniporter proteins because they're only able to move just one substance through. Now let's switch gears and discuss osmosis. So osmosis involves the movement of our water. We're now not discussing the solutes, which you see many solutes pictured over here. This is still a passive type of transport, and we are going to go through our selective permeable membrane here, which is our plasma membrane. So here we see our membrane is allowing water to move through, and it's doing this through a very special channel here called an aquaporin, or it could simply just move through the plasma membrane. Depending on how much water we have on each side and the solutes that are present will determine the movement of water. So let's continue that conversation. We have two ways that we moved through. We described this, how water molecules move through the plasma membrane or use these aquaporins. Another thing to consider are our types of solutes. So we might have some permeable solutes that we discussed in diffusion, such as oxygen or other nonpolar solutes, and we have non-permeable solutes. So for instance, we talked about glucose in facilitated diffusion that would fit into this, and they are unable to move through the plasma membrane on their own. So you can see these molecules are kind of being turned away here. So these would include also some charged particles, polar molecules, or large molecules. All right, now that we understand that, let's talk about our concentration gradients across the plasma membrane. So we have this tube here, and this dotted line is representing our semi-permeable membrane. We are going to see substances move through between the cytosol and interstitial fluid. So here we see the solutes cannot move, but we will see the water moving depending on how these solutes are distributed. So when we have a greater amount of solutes on one side, we will see water travel to that area until we have an even distribution. So here we see the black circles here are representing our solutes. The red and blue spots here, these are representing our water molecules. So we see that it's pretty even as far as the water distribution here, but we know that we have more solutes here on side A. Therefore, water is attracted to those solutes and is going to move through that semi-permeable membrane, and we end up with more water on side A. Now let's talk about osmotic pressure. This is going to be what drives osmosis, the pull of the water. And again, this is due to our solute concentration on either side. We will have If we have a steeper gradient, meaning we have a lot more solutes on one side than the other, then we will see that there will be greater osmotic pressure or a greater pull on the water to move to that side. When we talk about hydrostatic pressure, this is the pressure that's exerted by a fluid on the inside wall of its container. So we can think about this term in regards to our blood vessels and the amount of pressure that is exerted by that blood on the vessel wall. Now let's switch gears and talk about tonicity. So this holds hands with osmosis because we will see the water shifting in and out of our cells depending on the conditions. We are going to look at the change in cell volume and the osmotic pressure here. So when we use the term tonicity, it's the ability of a solution to change the volume or pressure of a cell by osmosis. So let's go through each of these solutions: an isotonic solution, hypotonic solution, and a hypertonic solution. First is that isotonic solution. So here we could see that the cytosol, the inside of the cell, and the solution surrounding the cell have the same concentration of solutes, and therefore we're not really going to have any change within the water volumes here. An example of this would be normal saline with a concentration of 0.9% sodium chloride. Again, usually we will find this in IV solutions and not have a net water movement. When we have a hypotonic solution, that means our solution has a lower concentration of solutes, whereas the inside of the cell has a higher concentration of solutes. That means we are going to have a greater pull of water and move inside of the cell, and that is going to cause an increase in volume and pressure inside the cell. That will if it continues, will usually lead to lysis and a rupturing of our red blood cells. And this is referred to as hemolysis, where our red blood cells will rupture. Lastly, we have our hypertonic solution. This is when we have a greater amount of solutes in our solution versus inside of our cell. That pulls out to the water into the solution, leading to our red blood cells to kind of shrivel up and shrink. This is also known as crenation, and you could see that in actuality in this image down here, in which we have a 3% sodium chloride water solution. In this situation, we'll see that we have a decrease in the volume and pressure inside of the cell, and this is incompatible with life. All right, now we are going to get into the differences between osmolality and osmolarity. You might have heard these terms used interchangeably, which is perfectly fine because there really is not a huge difference within humans. They are practically identical. There's only really a 0.5% difference between these. But before we start discussing those two terms, let's review what these other terms are and how they're measured. What are their units of measure? So a solvent is going to be the watery solution that we see over here, and that is going to be measured by either weight in kilograms or volume in liters. So keep that in mind because we are going to touch upon that when we discuss these terms. And then we have the solute. So we've mentioned this in the previous slides. This is going to be measured in weight, so in grams. And then we have this unit called osmol. This is the unit of osmotic concentration. It's our number of moles of solute that will contribute to osmotic pressure, also oncotic pressure of a solution. When we discuss these terms within humans, we will see that body fluids are typically going to be discussed as osmolarity, although in humans, we measure their osmolality first, which would be the osmoles of solutes per kilogram of the solvent, and then we translate that into osmolarity using calculations. And you'll see that most of our body fluids are typically going to be between 285 to 295 milliosmoles per liter. The range could be as great as 280 to 300. So again, just 5 milliosmoles per liter, pretty close. Okay, so now let's get into our actual osmolality and osmolarity terms. Our osmolality, the difference here is going to be that we are measuring the number of osmoles in weight. So we're using kilograms of the solvent. This is sometimes expressed in millimoles or osmoles per kilogram of a solvent. An example would be the grams of sugar that we dissolve in kilograms of coffee. Whereas osmolarity, the difference here is that we are measuring the number of osmoles in a volume. So we use liters of the solvent, and we can express this as millimoles or osmoles per liter. An example of this would be the grams of sugar into a liter of coffee. Now, I hope nobody is drinking a liter of coffee, especially at one time. But hey, we are in college here, right? And coffee is delicious. All right, and then just the same note over here. Okay, so as an example here, we have a, let's say, beaker of water. And if we were to add salt, our solute, to that water, what would happen to that osmotic pressure? If you want to pause and think, do so, because I'm going to reveal the answer. It is going to increase. So why do we care about this? It's clinically significant because it really helps us to assess our acid-base disorders and will assess any electrolyte disorders such as diabetes insipidus or SIADH, which is syndrome of inappropriate secretion of the antidiuretic hormone. This is also going to happen when you are really dehydrated and we start to lose a lot more water than necessary. So let's lastly do a quick little review of ADH. We have some osmoreceptors within our hypothalamus. So let's say we detect an increase in our osmotic pressure, like we said, if we are dehydrated. Then we're going to see the hypothalamic neuron here send a signal over to the posterior pituitary gland, and we are going to increase our secretion of ADH, and that will tell our blood vessels to vasoconstrict and tell our kidneys to increase water reabsorption, and that will lead to an increase in blood volume and an increase in blood pressure. So I didn't really talk about this node over here, but we also have some baroreceptors coming from the aortic arch and our choroid sinus. And so this ADH can also be released if we detect a decrease in our blood pressure. So that is it for this review. Please let me know if you have any questions.